Okay, so what I'm going to do is, so the idea now, so instead of giving a monolithic, lecture where I just go on and on about all the details of Gmatrix evolution, what we're going to try to do is I'll introduce some important topics briefly, so in the next like 15 minutes, and then each group will do an exercise using the Gmatrix simulation program to address a specific issue in Gmatrix evolution. And then each group will give a little presentation, so to kind of break up, the long speeches. Okay, so since about the year 2000 or so, we started working on using individual-based simulations to study the evolution of the Gmatrix. And the issue was, how constant are Gmatrices over evolutionary or ecological time, right? Because this had been a question that had been around for a while. And we've already talked a lot about the stuff on this slide. And so you already appreciate the importance of the Gmatrix and why Gmatrix constancy would be an issue, right? Because of course, if you're trying to do any kind of evolutionary inference involving the Gmatrix, you probably don't have the resources to re-estimate the Gmatrix every single generation, right? So if it's constant or relatively constant, or at least behaves in a predictable way over evolutionary time, then that frees you up to do a lot more analysis than you would be able to do if you can't make that assumption, right? If it was completely unstable, then effectively you wouldn't be able to do anything with the Gmatrix in terms of predicting the future of evolutionary responses or reconstructing the history of evolution for complex traits. All right, so we all appreciate the importance of Gmatrix stability. And in the year, around the turn of the millennium, I guess, the state of the evolution of the Gmatrix, there was sort of a debate about it. And so on the one hand, like it says on this slide, empirical comparisons of G between populations within a species usually produce similar G matrices. So at least they shared things like eigenvectors, leading eigenvectors and things like that. But as you moved out to higher taxonomic levels, you got more and more different G matrices, which kind of makes sense. The problem is that you can't really understand using analytical theory how stable G matrices should be over evolutionary time, because it's just not an analytically tractable problem. All right, so basically we were at a state where, one side of the argument was that analytical theory cannot guarantee Gmatrix stability, but then on the other hand, it can't guarantee Gmatrix instability either, and so we wanted to use individual-based simulations to kind of tackle this Gmatrix stability issue. All right, and at the time when we embarked on this multivariate approach to the evolution of the Gmatrix, there had been a history of using individual-based simulations to study a number of different topics in quantitative genetics, but they'd all been restricted to the univariate case, right? So it was just a matter of expanding those univariate models to the multivariate case. All right, so we wanted to use these same basic models, but use them to study multivariate evolution and the evolution of the Gmatrix with at least two traits. All right, since then, which I'm not going to really talk about, we've gotten more interested in the evolution of the Mmatrix, because it turns out that the Mmatrix plays a really important role in determining Gmatrix stability and the form of the Gmatrix. All right, and then more recently, which we probably won't get into at all, we've started looking at how epistasis can allow the Mmatrix and the Gmatrix to evolve. All right, but what I'm going to do for the next couple of minutes is just tell you about the model and how it works so that you kind of understand the model, and then how to use it, how to use the simulation program. All right, so basically it's a Monte Carlo simulation that keeps track of every gene and every individual in the population. We assume a certain number of loci that affect the traits, and all the loci are pleiotropic in this model, right? So if you wanted to relax that assumption, it could be done, but not within the model as it's set up right now. Everything's additive, which is nice and simple, and so if everything's additive, then the total genetic variance is the additive genetic variance, so you can directly get the G matrix based on the individual's breeding values. Allelic effects are drawn from bivariate normal distribution whenever there's a mutation, right? So this is a very important part of the model. So there's going to be a mutational distribution that describes the mutational effects of new mutations entering the population. And it's really important to understand now that every allele, since every locus is pleiotropic, each allele affects both traits, right? So we're only going to do a two-trait model, but each allele affects both traits, which means that when there's a mutation, it could change the allelic effects on both traits, right? So the new allele is going to have new allelic effects on both traits. The change in the allelic effects is drawn from a bivariate normal distribution. So those the change in allelic effects, those could be correlated, so that when you get say there's a positive mutational correlation, then if you get a positive change in trait 1, you'll also tend to get a positive change in trait 2 whenever there's a mutation in an allele, right? This can be a little bit hard to think about because we're not really used to thinking about these bivariate cases for particular alleles, right? But you can imagine that if two traits share similar developmental pathways, then a mutation in a gene in that pathway could have a similar effect on both traits. That would be a positive mutational correlation. Does that make sense? That's a really important concept. So if you're confused by that, everything else will probably be confusing. So yeah, there's a picture that I'll show you in a minute. So there's a mutation rate. It's pretty high because you have to have high mutation rates in these kinds of models to maintain any genetic variance in populations. We draw environmental effects from a bivariate normal distribution with a mean of 0 and variances of 1. You can kind of think of that as the scale of the model. We have a Gaussian individual selection surface with a specified amount of correlational selection and different values of omega that you can change in the model. Usually we use 9 or 49. Larger values of omega are weaker selection, right? So if you have a larger value, it's like having a larger variance. It's a variance-like term, which makes a shallower surface. And so then you're going to have weaker selection. And then each simulation run might run for a few thousand generations at the beginning to equilibrate. And then we would have experimental generations where we keep track of the variables of interest. All right, so here's the basic life cycle, right? So you start with a population of n adults. They produce progeny. In this basic model, they're monogamous. We have regular rules of Mendelian assortment, and we have mutation and recombination at this point. All of the loci are physically unlinked in this model, so there's free recombination. You could still generate linkage disequilibrium, though, due to things like selection. All right, and then they produce a certain number of progeny, so B times N, where N is the number of individuals in the population. Then we have a period of selection, which is imposed by that Gaussian selection surface. They're going to be greater than N survivors. If they're fewer than N, so N can be thought of as the carrying capacity. In these simulations, if the population drops below the carrying capacity, it just ends the simulation. And the reason is that we don't want fluctuating population sizes to be a factor in affecting the evolution of the Gmatrix, right? So basically, we just terminate a run if it drops below the carrying capacity. And in general, that means that we're using fairly weak selection so that many more progeny are produced so that there are many more survivors than the carrying capacity are typically produced. All right, in the old simulations, we would start with a population with no genetic variation at all and run it for 10,000 generations to get to a mutation selection drift equilibrium. In the simulation version that you have, it starts with some genetic variation to make it a little bit faster, so you don't have to wait around for 10,000 generations every time. Then we would impose a desired model of movement of the optimum. So basically, all the selection is imposed by moving the optimum. It might stay in one place, then we'd have a stationary optimum and have primarily stabilizing selection. Or it might move, and that would impose directional and stabilizing selection depending on the position of the phenotypic distribution relative to the optimum. So beta is not a parameter in this model, right? This is just, beta would just emerge from the model as a consequence of the mean being displaced from the optimum. The Gmatrix also is not a parameter in the model if you think about it, right? Because all we're specifying are allelic effects and mutational effects and things like that, and then the Gmatrix emerges from those various population level parameters. All right, we focused mainly on average single generation changes in the Gmatrix because we were interested in how the different parameters of the model affect stability of the Gmatrix. All right, so like I said, the Mmatrix is really important. It'll turn out for the evolution of the Gmatrix and the stability of the Gmatrix. And so basically you can think of the Mmatrix in kind of the same phenotypic space as the Gmatrix. And you're going to have mutational variances and a mutational covariance. And if you have zero mutational covariance, then every allele, when it mutates, will still get two allelic effects, but they won't be correlated, right? So it could affect trait 1 in a positive way, or trait 1 in a negative way, or both positive or both negative with equal likelihood. The alternative is to have a very strong mutational correlation, and it could either be positive or negative, right? And so then that means that if you have a mutation at an allele, you're going to get mutations affecting both traits. But if you have a positive mutational correlation, then if trait 1 is affected in a positive way, then so will trait 2 be affected in a similar way. Does that make sense? And then a negative mutational correlation would mean that if trait 1 is affected in a positive way, then trait 2 would be affected in a negative way by that individual mutation. All right, and you just talked about selection surfaces a whole bunch, so I don't really need to go through this. But we're going to be operating in omega space for our selection surface, right? So we're going to use omega to specify it. Like I said a minute ago, larger values are weaker selection, smaller values are stronger selection. And then you can also specify selection, the strength of correlational selection by changing the off-diagonal elements. Okay, and in the actual simulation, you set the selection correlation rather than the covariance because it's just easier to think about. Yeah. They're just magical, arbitrary, theoretical units, right? So you can sort of think of them in terms of the environmental variance. And so basically, if you think of them in terms of phenotypic standard deviations, that's sort of the units that they're in, right? So the phenotypic standard deviation in these models is going to be around, the heritabilities will probably be around 0.2 depending on the population size, which means that the phenotypic variance will be something like 1.2, right? So 49 is quite weak selection, but it still has very strong effects on the population. Okay.
So if you think of the with this kind of a selection surface, if you think of the phenotypic distribution as having a standard deviation of variance of 1, then this has a variance almost 50 times larger. Right? The spread is really substantial. So that's one way to think about it. And that's like the coefficients in the P-matrix, right? They're bigger and the whole thing.
All right, and then in the simulation, so we've already talked about the G-matrix too, and that you can sort of draw an ellipse around the breeding values to get the G-matrix, where 95% of the breeding values would be within that ellipse. In the simulation, it's only going to draw it's only going to allow you to have 2 traits, right? Because that's all you can visualize easily on a computer screen, and then it'll draw the 2 eigenvectors, right? Scaled by the eigenvalues. Right, so basically it won't draw the ellipse around, but you can imagine an ellipse around the leading eigenvector and the second eigenvector, right, as describing the G-matrix. Okay, and the variable that we focused on mainly as a measure of G-matrix stability was the angle of the leading eigenvector. So you could debate whether or not this is the best way of measuring G-matrix stability, but this is the one that we chose. And the reason we chose that is that the orientation of that leading eigenvector is a major constraint for evolutionary trajectories, right, which you'll see when you run the simulations, right? So if you have a very eccentric G-matrix that's oriented at some arbitrary angle in space that deviates from the direction of selection, then you're going to get a response to selection that's highly constrained by the orientation of that G-matrix. And that's why we were most interested in the angle of the G-matrix rather than other things. There are some other aspects that we measured, like the total variance and things like that, which are also of interest. But the main thing that we focused on was the angle.
And so sometimes that's called, the angle is called Gmax. So it's the direction of maximum genetic variation, measuring the change in Gmax per generation for stability.
Yep.
Is that it?
Yep. All right, so that's kind of all the introduction that I'm going to give on the underlying theory, and now I'm going to talk a little bit about how to use the simulation program, and then we'll break into our groups and try to tackle some of the questions that are in the assignment or the exercise. All right, so if you're running these kinds of simulation type models, there are a couple of constraints you run into that are a little bit different than empirical studies and a little bit different than analytical theory. And one is that they generally have a lot of parameters. And so you really can't do a fully factorial approach in your experimental design. You can't really vary every parameter in combination with every other set of parameters, because you would just you would have so many different combinations of parameters that you could never run them in your lifetime. And so it's better to think of it in terms of experimental design, where you're thinking about controls and treatments. And so you would start with kind of a core set of parameters that you hope is sort of realistic and sort of represents what would happen in real systems, and then change a couple of parameters at a time and see how those parameters affect the system, right? So you would change those in tandem when you think that there's likely an interaction between them, and then some you might just change on their own to get an idea of what they're doing. But you can't really realistically do a fully factorial approach. The default values that the program's already set at are a pretty reasonable starting place, but when you start, you probably want to remove any kind of stochasticity and use a stationary optimum as your starting place. And you might want to increase the curvature of the selection surface to 9 instead of 49, the default, because stronger selection might give you a more dramatic result that's more obvious. All right, so this slide is mostly so that you can look at it later, but I'm going to go through all of these. But there are a couple of things I want to point out. So one is, if you reduce the number of loci to a really small number, then it'll run faster, but if you reduce it too much, you'll lose all genetic variation and the program will crash, right? So at some point, if you have no genetic variation, it just crashes, because some of the calculations aren't possible with an absence of variation. If you change the selection surface, there are actually two places to change it. So change both places. One is before the optimum moves, and one is after the optimum moves. So in some cases, you may just have the optimum move once, and you could change the selection surface so that it's different after the move compared to before the move. That's why there are two sets for the strength of selection. There are two sets for the selectional correlation. And if you change one in general, you want to change the other one just to keep the same value before and after the optimum moves, unless you're specifically interested in how a change in the orientation of the selection surface would affect the G-matrix. Right? That might be something you're interested in. This parameter right here tells you how far the optimum is going to move. So how far will the selection surface move each time that it moves? And so if you want a stationary optimum, you would set those to 0. If you set them to a larger value, then the optimum will move as the simulation runs. All right, so I'd start at 0, and then having it move, I would start at a very low value if it's a per generation move, because if it goes too fast, the population will go extinct. But you'll find out right away, because it'll just crash the program ultimately. Actually, I think it'll finish, but it'll end prematurely. All right, and then this down here, this parameter right here, introduces stochasticity in the position of the optimum. So basically, this is the variance in the position of the optimum. So if you make these non-zero, then every time that the optimum moves, you'll also draw random numbers from a bivariate normal distribution and move the optimum in some random direction based on those values. And then down here, you can either move the peak once or move it repeatedly. If it's stationary, it doesn't matter which one of those you pick. But if you have the optimum move, if you choose once, it'll just move once at a specified generation, which is right here. You could make that 100 and it would move at generation 100. If you move repeatedly, then it'll move every single however many number of generations you put in here, right? So for these parameter combinations, the optimum would move every generation by 0.01 for trait 1 and 0 for trait 2, right? So whenever there are two of these, the first one is for trait 1 and the second one is for trait 2, right? Hopefully the rest of this is self-explanatory and you can look at this as you're looking at the parameters of the model. Okay, and we're here if you have any questions about the parameters and how to set them. All right, so then here's how you actually run it. It's actually pretty simple. So this little arrow right here runs it. So once you've chosen your parameters and hit OK on the parameter window, then just click that button and it'll run. At the end of the run, if you want to save everything in this window that appears, just hit this button and you can save it as a text file or whatever it'll save as a text file. This is the button to get the parameter window to pop up so you can set the parameters. This button just causes the graphics window to appear and disappear, so don't worry about that right now. If you want to save every individual run in its own file, check this box right here, okay? And it'll save, as it runs then, whenever it finishes a run, it'll save that run in a comma delimited text file that you can import into Excel or R. Does that make sense? And so when you check the box, it'll ask you for a path where it's going to save those files. And then if you want to change the path, just uncheck it and check it again.
What if you want just the averages of all the runs?
It'll save those in a different file in that same directory, right? So it'll also save the average. There'll be another file in that directory that has the means for each run and then the grand mean across all the runs for all the parameters that it outputs. Yeah, I think it's means and standard deviations or something. All right, and so then as it runs, there's also a graphical output. So if you have it draw the Gmatrix, if you put in 0 for how often to draw the Gmatrix, then it'll be an animated output where the dark lines are the current Gmatrix. So you'll see it evolve as it goes along. So this is a good way to get a feel for how the parameters affect the Gmatrix. And then it leaves kind of this gray trail where the Gmatrix has been in the past. And then the red cross is the position of the optimum, right? So in this case, this Gmatrix is kind of eccentric and it kind of chased this optimum, but it ended up, and then the x-axis is trait 1 space and the y-axis is trait 2 space. And so it ended up displaced, even though basically the optimum only moved to the right, trait 2 still ended up displaced from its optimum due to a correlated response to selection there. And then if you want specific G matrices from particular generations, you can have it draw the G matrix every, say, 100 generations. That's one of the parameters. And then it'll just draw that G matrix, at generation 100, and there's the optimum that goes with it. And then all the little blue points are the breeding values of the individuals in the population. And then this would be generation 200, 300, 400, 500. So if you want snapshots of what the G matrix looks like as it's running, then you can set it to draw it every however many generations, 250 or 100 generations or something like that.
While we've got this up here, this illustrates the flying kite
It does.
result, doesn't it?
Yeah. So whoever has the flying kite effect, this is what it is.
Imagine a little string from the optimum to the G matrix, just like a flying kite.
And so as the optimum runs away, even though only the trait 1 optimum is moving, because there's a genetic correlation, trait 2 is getting displaced from its optimum because there's such strong selection on trait 1, right? And then you get a correlated response to selection on trait 2.
Using that technical term we introduced, you could call this perpetually screwed.
All right, so the last thing, one thing that I forgot to mention, if you want to set the number of complete replications of this particular set of parameters, this sets it. So if you just want to try one run with a particular set of parameters, set that to 1 if you just want to see what's going to happen. But then if you want a bunch of replicated runs, set it to a higher value to get 5 or 10 or something like that. All right. And then the meaning of the variables is in the variables that it outputs. They're pretty self-explanatory, but they're all described in the documentation that comes with it. And so basically you have the position of the optima for trait 1 and trait 2. You have, the mean trait values. You have the phenotypic matrix. You have the Gmatrix. And then you have the angle of the Gmatrix and the eigenvalues. And then the single generation changes in these, which are either going to have a D or a little squiggly line in front of them. And it's just 2 traits. Yeah, you shouldn't be able to change the number of traits. And if you could, it would just completely crash. I mean, it's not built to withstand 3 traits.